Hello, welcome to today's discussion series. We'll give a minute here for folks to filter in. Thanks for being here. Excited for the conversation today. As you're filtering in, feel free to throw into the chat where you're tuning in from. We typically have folks from all over the world. Sometimes Zoom does kind of a weird thing where the chat defaults to only chatting with hosts and panelists. So if you see that, make sure to hit the drop down and chat with everyone so everyone can see where you're coming from. Welcome to today's discussion series. The Singularity discussion series is a monthly webinar that we host with various experts covering the breadth of the taxonomy of what we cover at Singularity, from technologies to application areas. Super excited to have Dr. Eriona Hysolli here today with us. And in a moment here, I'll pass it over to her to introduce herself, and then we'll dive into the conversation.
Okay. Is that my prompt to start my intro?
Yeah, you know what? Let's go ahead and get started. I think we have a quorum here. We have folks from Abu Dhabi, Taiwan, Colorado, South Africa. This is amazing. So yeah, I think folks will continue to join, but I think we have enough folks to get started. So welcome. My name is Cody Hansen. I'm on the Singularity team. I'm the host of this monthly discussion series. And yeah, as I said, today we have with us Dr. Eriona Hysolli. Eriona, you want to introduce yourself and kind of share a little bit of your background and what you're up to?
Yeah, absolutely. I'll keep it short because I think throughout the discussion, I think probably a lot more information will come out. So I don't think we have to cover right at the beginning. But hi, everyone. I really would like to thank the organizers, Cody, definitely, and also for the interesting mini discussion we had prior to this session with Cody and Venus. So thank you so much for the invite and the opportunity to share some of my thoughts. I actually want to thank Singularity as well. This is sort of like the second event I attend with them. I was in San Diego at the Future of Biotech event, courtesy of Alex. She invited me and I had a blast. And so I hope to, participate more in these sessions in the future. Very, very exciting to always look forward to what we can do in technology. So I'm Eriona. I did my PhD in stem cell biology, and I think that will be also relevant to some of the discussion later on. And then I went on to do a postdoc working on de-extinction and radical genome engineering in the laboratory of George Church, who's a pioneer synthetic biologist. And through his lab, I was also able to join the startup that most of you are familiar with, Colossal. George has been a pioneer of de-extinction as a concept to reverse some of the ecosystem damage we've done with loss of species. And so can we reverse this trend? We are losing biodiversity at alarming rates. So is there something we can do leveraging the technologies of today and those of the future in order to reverse these negative trends? So I was very, very fortunate to work on the project of the woolly mammoth de-extinction and a lot of the technologies that are needed in order to make this project complete and successful. So, I'll be touching on some of those technologies as well throughout this discussion. I've been very, very interested in, some of the future work we need to do as a species to reverse some of the negative trends that are affecting our own species, such as low fertility and replacement rates. And so hopefully we'll touch on those salient points through this conversation. And so super looking forward to. Thank you again for the invite. I do have to say, I want to put a disclaimer out there. A lot of the things that I will be talking about are also personal opinions. Some of those are shared by others broadly, some of them maybe more sparingly, and maybe some of them like not shared at all. I think my mission is to keep the conversation open-minded and transparent. Usually as a society, we move towards a certain direction through these collective conversations, but these ideas should be, any of these ideas should be discussed nonetheless. And then so like we collectively move in certain directions as a society. So just be open-minded, regardless of what you think about sort of like the topics of discussion. I think it even in sort of like engaging in them in a more philosophical manner can be quite fun.
Thank you for that. And yeah, this obviously can be, I think, maybe a touchy subject in some ways, and it's obviously very, very innovative. So keeping an open mind and operating with, assuming positive intent is, I think, a really important note. So can you share a little bit about what took you from your work at Colossal and de-extinction into this interest in human fertility, and what connected those fields for you? And then also on top of that, um, what is some of the work that you were using in de-extinction that has proven most valuable in addressing human reproductive challenges?
Yeah, so I'll walk back a little bit towards what motivated me throughout sort of like my scientific career to date. I've always been interested in sort of like these topics that are very forward looking and maybe take a little bit of time to either fully realize or fully be more accepted. But of course, time shouldn't be a deterrence to some of these very, very important things to discuss for the future. And so, yeah, I've been motivated. For example, what spurred me to go into the stem cell, at least induced pluripotency and embryonic stem cell work, is the importance they have, not only in sort of like developing to the best of our abilities, human health and disease and development, but completely and potentially eliminating these proxy species we use or animal models for studying such topics. I think obviously the best model to use for human-related work is the human model. So stem cell technology offers that potential. Now, of course, a lot of that potential is in the process of being realized, but will be there. It's all feasible. So eliminating completely animal models and animal testing, I think that's actually what motivated me a lot to do the PhD thesis I did. And then, I was thinking a lot as a grad student as to what makes humans humans. And I think that that stayed in the back of my mind, but also definitely played a role in reaching out to George and joining his lab to work on the woolly mammoth de-extinction project. While there, I also worked on technologies. Obviously, you do need very, very important and crucial technologies to make these important, ambitious projects happen. But the concept of reversing extinction, I think that was, um, you know, quite interesting to me. And I think that can obviously be a game changer for our biodiversity crisis that we're currently in. So, I was fortunate enough to sort of like work on on mammoth de-extinction, and the company obviously is interested overall in species de-extinction. But so while working on this topic and while also being confronted with some of the bleak statistics when it comes to human species themselves with low replacement rates, in places like China, it's around one, it hovers around one. In Europe, it's usually just between 1.2 and 5 births per woman. And even in the United States, for the past few years, it's been at around 1.6, and the average should be 2.1 or above. So when we talk about extinction, generally we talk about other species extinction, but is it time to take it more seriously that we could also face our own extinction if we don't reverse such negative trends? So that's sort of like what's in the back of my mind as I consider what are some of these reproductive technologies of the future that we can use to reverse these negative trends. I think philosophically, it's also important to discuss the topic of, like even in the best case scenario, as a species, does that mean that we really have to adhere more to these innate, primitive, biological needs? And that's an interesting conversation to have. Maybe as a species, even when all of the ideal technologies are there, to carry forward more progeny, will we take it? And I think it's an individual choice. We don't necessarily have to make a decision for everyone, but I think as a species, I think thriving in the future is still very, very important. We can all agree that despite all our faults, we're fairly unique, and keeping this unique flame going, I think it's actually imperative. So yeah, those are some of the things that have motivated me. So in a way, it makes sense working on these technologies for reversing extinction in other species, but can be very, very relevant to potentially reverse our own extinction.
Yeah, so diving right into that technology, let's talk a little bit about in vitro gametogenesis. Can you explain in layman's terms what it is, and why it might be a game changer for people struggling with infertility?
Yeah, absolutely. So the big picture is like, of course, we want to think about the thriving human population, but on sort of like more practical day-to-day terms, there are a lot of people who just really want to have children that might not be able to because of age, because of sort of like reproductive infertility, because of, in some cases in same-sex couples, unless you use some of these assisted reproductive technologies, you won't be able to have them. So you want to sort of like democratize if you really want to have children, you should, and we should use all of the technology out there. One of them is this very, very cool concept and work on in vitro gametogenesis, which I can quickly explain, and I can do a deeper dive or not, depending on sort of like the interest there. But in our body, of course, we produce germ cells, which are the cells that go on and mature to form the egg and the sperm. That gives rise to embryos later on after fertilization. So of course, so far, at least until more recently, you can only produce these sort of like cells in the body or from the body. But they are at least in the case of female reproductive prowess, usually there are like limited supply, so to speak.
Hmm.
So if you're playing your choice to have children, or you are unable to, or there are some of these infertility conditions that afflict women, such as because premature ovarian infertility and low ovarian reserves and so on. So can you potentially make an unlimited supply? And of course, it's not just biological for some of these sort of like low fertility rates, of course, societal and environmental factors, but at least tackling the biological genesis, can you make these germ cells in a dish? And so actually, it's been tremendous fundamental work by Japanese groups, especially where they've been able to produce live mice because they were able to reconstitute or recreate the germline or germ cell making machine outside of the body, outside of the mouse body, and then give rise to live births. So that's very exciting because some of the preliminary work has also been done in humans. Obviously, there's a little bit more work to be done in terms of making functional. The ultimate test is, can you produce a human, or can you produce an animal from these cells? Not just having the hallmarks of germ cells, but actually producing, the live births of whatever animal is in question. So yeah, so that's fundamentally what in vitro gametogenesis is. And there are a few companies out there that are attempting, early stage companies that are attempting to make human sperm and human egg in a dish. So like really inspired by these fundamental Japanese group that did a tremendous work in mouse. And of course, you can be kind of a little bit fancy with it, because these technologies also allow things like bipaternal progeny. For example, some of these Japanese groups have produced live mice from germ cells from two dads, pretty much.
Hmm.
So that's particularly relevant potentially to male same-sex relationships. So very, very exciting work. Again, I think definitely we need to do a lot more work on sort of like the human side, but being able to do this very easily in unlimited supply outside of the body in a dish, and to also use preserve for the future, so you're not necessarily constrained by the timing when you want to have children in the future. I think it's actually quite exciting technology. And as I said, proof of principle has been done. It's a matter of trying to carry the success in mice to humans, and thankfully, there's already massive efforts on that.
And what types of cells are used, or does it not really matter? Any cell from the body can be used to create those.
Yeah, that's great. So in terms of where you want to start with a multi-potential, pluripotential line. That's why a lot of this work starts with a pluripotent stem cell or an embryonic stem cell.
Okay.
But thankfully, because of the induced pluripotency work, also courtesy of like a massive, Japanese stem cell biologist, Shinya Yamanaka, who was able to recreate the process of making embryonic stem cells in a dish artificially using transcription factors or genes, a combination of genes. So he obviously was a pioneer of the technology called iPS technology. So you can actually start really, really go back and start with any cell in the body, make these induced pluripotent stem cells that are very, very similar, almost identical to embryonic stem cells. And then those can be coaxed either through media conditions or through a combination of genes or transcription factors in order to make these germ cells. And then, of course,
Interesting.
It's a little bit more complex because you need to mature them to be ready for fertilization and so on. But pretty much every step of that has been sort of like uh, um, proven fundamentally or conceptually in animal work.
Interesting. Yeah. So there's a convergence happening within this that connects a broad range of possibility there. Um, so along similar, but I guess even more dramatic lines, synthetic embryos have been making headlines recently. What's your perspective, opinion on what's actually possible today? What's science fiction, and what are some of like the major ethical concerns of those?
Yeah, tremendous important question of today because there's really some very elegant, beautiful work that's coming out, at least early stage, right? Where we're not ready to strike, potentially tackle the later stage development. And of course, there are some fundamental challenges that need to be overcome in order to continue development further. But early stage, these are called, there's like a variety of names. You'll probably hear me, mentioning a few of them. There's obviously the synthetic embryo, but in sort of like in the scientific terms, they're called blastoids. So there's, I mentioned those embryonic stem cells before. They are truly magical because they can give rise to anything, including like a full body, a full organism, a full human. So there was beautiful work done on the mouse. As you know, a lot of the cool work starts in mouse, and then sort of like you
Mhm.
go up from there. Where a great Israeli stem cell scientist, Jacob Hanna, he was able to develop mice at early stages of development, but the furthest that's been done to date from just starting just from these embryonic stem cells or induced pluripotent stem cells. And that's quite exciting. So you can start there and then sort of like have an early stage developed embryo. So of course, you're trying to recreate that kind of process on the human side as well. You can do it through different ways. Either you take these embryonic stem cells into a more naive state, which is a little bit more backwards, where they have the potential to give rise to the trophectoderm layer as well, which is very, very important, later on for development. Or you can actually sort of combine different lineages together. They aggregate and they form these embryos that really look very, very similar to what you see in vivo, whether mouse or human embryo. So that's very exciting work. Now, as you can imagine, with some of this work, there are ethical implications. So far, there's that 14-day moratorium generally, plus or minus. In places like the US, it's more of a guardrail. It's not a law. So there's ways to push that a little bit. I think there's an American group that pushed the formation of these early stage embryos, synthetic embryos, blastoid, interchangeable to around 21 days, which is very exciting. Now, of course, as I mentioned, there is a little bit of a challenge to go further, even without the guardrails, because you do need a network of vascularization or blood exchange network, so that these aggregates can grow bigger and bigger, right? Without
Mhm.
that, it's very, very challenging to grow them bigger. That's also something that even in these organoid space of stem cell technology, you're limited by diffusion. So you do need the vascularization network to make this development proceed further. But I think that's super exciting that, if allowed, you can really try to push the limits how far they can go. And the reason for that 14-day window is, generally speaking, that's when the body plan of the embryo starts taking shape in this process called gastrulation. So it's kind of like a little bit, I mean, it's obviously rooted in biology, but it's also a little bit of a, just a particular cutoff. I think we can push that a little bit further for sure, especially if we want to know like how good we can make these blastoids, these synthetic embryos. But yeah, apart from the biological challenges, those are some of the moratorium that a lot of the scientists still adhere to, and so they don't push them further than, let's say, day 20 or so. So that's kind of like the limitation.
Yeah, and this ties in nicely, I think, to the next question, which is, we're starting to hear about artificial wombs. And are those how close are those to being real? It sounds very much sci-fi. And what types of technologies still need to be invented for that to be real? And then I also, I know a lot of this is your opinion on the ethical side too, right? Because everyone has a different level or gauge of what they're comfortable with. But just curious, what's being discussed in the scientific community right now around the possibility of artificial wombs and the ethical considerations of artificial wombs.
Yeah, through sort of like what's also openly discussed versus what also some of the things that are sort of like brewing in the background.
Mm-hmm.
So a space that's heating up quite a bit. And there is obviously a biological foundation to doing that. And it does sort of like depend very much on some of these early, even the synthetic embryos and in vitro gametogenesis work, just extending that completely. So can you extend the full developmental stages to doing that in vitro or outside of the womb ex utero, right? There's like no reason, no like fundamental reason not to do it apart from like discussing the ethics of it. So just to complete the full process outside of the human body. And so where are we now? In terms of what's feasible, I mentioned the work on the mouse. You can take it to embryonic day 7.5 or 8.5, 4.5. I think there's some new technological advancements that push it a little bit further. That's probably going to come out soon. Yeah, in my opinion, beyond just gestating humans in the future completely ex utero, that can be, to counter infertility crisis, also, going from a colossal mission, also was interested in using these kind of technologies for conservation purposes, so that you can, rewild animals, but without having to necessarily burden the endangered species for restoring the health of their own species.
Mm-hmm.
You can really model a lot of the diseases as well using these kind of ex utero developmental models. So like, can you really model preeclampsia in a dish, right? So can you study the roots of the disease and what can you do about it? Or
Right.
PCOS, no, like obviously can be used to completely remove the limitation of age when you want to have a child in the future. And a lot of the embryos fail because of chromosomal aberrations like super early on. So can you use this kind of devices in the future where you can gestate multiple to find the right one that can be fully carried to term? So again, so like whatever you think about what that means for the future, I think the foundations of that are being laid right now. Early stage development, obviously in progress. And as soon as we sort of like have a more open discussion of where the cutoff should be potentially for full gestation, I think that's where we should be heading.
Right.
Having said that, there are, as I said, biological challenges that are being overcome, even in sort of like in the best case scenario. Let's say that there are no guardrails whatsoever. How do we mimic placentation, right? How do we, what sort of like biomaterial should we use for the embryo to sort of like invade, just as it invades in the uterine wall? So those are
Mm-hmm.
some very complex processes that need to happen. The embryo has to access the blood source. And so it does that through like this process of invasion. And so can we mimic that outside of outside of the body? There's definitely a lot of cool fundamental biological work to be done there. But again, without sort of like actually doing it, you don't really know. And so I think that's why I'm sort of like a big fan of taking that work, especially with some of these proxy here, I don't mind at all. Also, like really relying on these proxy species, developmental processes, even for more, even for species like mouse can be very, very complex. So we can learn a lot. And I'm actually very happy that that's being taken the furthest it can be taken step by step.
Right. I do want to call out here for folks that are listening. If you have questions, please feel free to throw them in the Q&A tool or in the chat. We have Venus on our team behind the scenes curating those, and we'll jump into audience Q&A on the back 20 or so minutes of this 1 hour conversation. So, but in the meantime, I'll keep going forward here with my questions. Diving into less future focus and more about like today than say the next 5 years. For someone dealing with infertility right now, what are some emerging technologies that might realistically help them in the next 5 or even 10 years?
Yeah, I mean, you probably are all very familiar with IVF. That's been sort of like a game changer ever since the first like IVF baby in the UK was born, Mary Louise Brown, 1978 or so. And it's exploded. I think it's estimated that around 10 to 13 million babies have been born via IVF. Now in the US, 1 in 42 are born via IVF. And of course, in places like Spain, which is very interesting to me, almost 15% of births are through
Wow.
IVF. It's very, very exciting to see as well, because it can really be life-changing for a lot of people. So even that is a process that's very much still done manually, right? With low success. So what can you do to push the efficiency, make the invasive process a little bit shorter? And of course, there's actually companies in the space to try to shorten the IVF cycle time to mature or to shorten sort of like the process women have to go through for IVF. I think that's actually a very important work to be done. I want to see more automation, of course. I want to see better screening. So can we have companies, of course, that can screen better for these chromosomal aberrations or for diseases? Some prenatal screening is done, but can you push the limits to how many, loci you can test, how many genes you can test for aberrations? I've seen some rejuvenation work being explored. One of the things you can do in a way that kind of takes care of um, rejuvenating the reproductive organs is rejuvenating the whole body. And you probably are familiar with
Hmm.
some companies in the space that are trying to do in vivo reprogramming. So using that same process that made these iPS cells, these embryonic stem cells, but in lower dose throughout the body for full body rejuvenation. You probably have heard companies like Altos, New Limit, and so on. So that can, in a way, if used preventatively, be used for sure, like more rejuvenation across the board, including the reproductive organs. Uh,
Right.
that's actually quite exciting to see how that might affect the reproductive organs and their biological age with these rejuvenating techniques. A mitochondrial transfer transfusion, when they become sort of like more tried and tested. I think that could be very, very powerful. More immediately in the next few years, probably it will take off a little bit more. You probably have heard sort of like this technology of platelet-enriched plasma that actually has been used successfully to, at least in some cases, to increase, for successful births, even at a more advanced age for women. So I think fundamentally, and what also I'm interested in is, can you actually do more work in vivo rather than in vitro as well, in terms of
Hmm.
treatments, uh, all kinds of treatments, including substantial drug development to treat some of these root causes of biological infertility without necessarily always relying on the in vitro and exudaral work, which will happen anyways. But I think it's also good to tackle it from can you do something, whether by a drug or by a treatment or by therapy to treat some of these root causes of infertility in the body in vivo. So as I said, PRP, mitochondrial rejuvenation, in vivo reprogramming, so those are some of the things that are being explored in one way or another. But even the process of this very true, tried and true assisted reproductive technology called IVF, making that process more efficient. As you know, like in a lot of clinics, you're relying on a human hand to do this, and that can be
Right.
with different levels of expertise, and that can be quite challenging to like standardize that. So can we make that more, can we use sort of like robotics and make that more automated in the future, which has not happened yet. So those are some of the things to look forward to, more therapies that can be adopted even like more short term in the next like 2 years, if not already adopted.
Right. Yeah. What's a good research or information source for folks that want to stay on top of this stuff?
I mean, I can compile, for example, after this, I can compile sort of like a resource as well.
That'd be awesome. Yeah, we can share that in the show notes afterward via email.
It is a little bit of a cheat sheet, but definitely use some of the sort of like chatbots, AI chatbots, of course, to find some of this information. It
Yeah,
can be a good summary for you, even insofar as like digesting some of these primary research papers. So I highly recommend some of them. But I can also sort of like compile a list, at least an intro list for the people who are interested.
that'd be amazing. And we'll share that out within a couple of days after this conversation. Um, from an investor or an entrepreneur, company builder perspective, what's the most promising near-term commercial opportunities do you think in reproductive biotech?
Yeah, I mentioned some in this like in the previous prompt, but definitely some of these rejuvenating in
Hmm.
vivo reprogramming. I think they have a lot of promise, and so definitely you'd probably see some of that fundamental work be released more and more in the coming years. So, and definitely this is not a one group will solve everything. Definitely more investment across groups, across teams will actually increase the probability of success. So still, in addition to sort of like IVG and ex utero, which are still in sort of like in the genesis, some of these some of this work that's already ongoing, it can really do with more investment across the board, so that people work in parallel. There's in a way fundamentally more than one way to skin a cat, right? So you do
Right.
democratize the investment in this space given its importance, but also I think something that I mentioned again, but AI-driven drug treatments for infertility or in vivo treatments, whether gene therapy, cell therapy, like can you actually, either transplant um, ovarian organoids into senescing organoids to make more? Or can you treat such that there are more follicles to produce, that ultimately produce the more mature oocytes in vivo in the female body? We should explore more of these like in vivo technologies, and I think there should be more investment in the space. Interestingly, this is something that interests me, of course, the in vivo aspect of the work, but in some cases, you probably know with it's a very low incidence, but the incidence of multiple pregnancies, like triplets, multiplets, and so
Hmm.
on, there's a genetic component to that. A lot of women hyperovulate. There's a genetic component that you probably have heard to having multiple pregnancies. So actually, I'm interested in like understanding the genetic component, the true genes that make this happen, so that we can offer treatment. Like, can you potentially release more than one egg at a time, which is what happens with some of these multiple pregnancies? And can you do that in such a way that it really increases the chances of pregnancy? Because it's like usually there's one shot, right?
Right.
Every month. But like, can you have multiple shots on goal, so to speak? So
Right.
that would be it's an area that I haven't really seen much being explored, but I'd really like to see. You really having women, release more than one egg monthly on the regular basis to increase, the chances of a successful pregnancy. That would be something that will be very cool. Increasing the number of, I guess, multiplets. I think that would be pretty cool.
Yeah,
Again,
that's it.
opinion. I don't know whether everyone will be on board with that.
No, but it's interesting and it's cool. I mean, you kind of obviously perked up there at that idea and thought. So it's obviously something that interests you and is very cool to see. Um, on that note, what scares you, keeps you up at night around all of this technology, and what gives you the most hope?
Uh, Yeah, so what keeps me up at night, interestingly, is more humans losing their competitive edge in the age of AI, right? So I really want
Mm,
for humans to thrive in this new age we're entering. We've probably entered,
right.
So we are definitely, as I mentioned, we're definitely a very unique species. So I want to, think about all the things that could preserve our future. So yeah, but I'm still very, 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 very hopeful and excited. But that's what keeps me up at night. But in terms of like what I'm excited about, all of the technologies we're talking about, right? I think we're on the right path to making a lot of them happen. And I think it's cause for optimism.
And how do we make sure that these technologies, as they get kind of unlocked, benefit ideally everyone equally? I mean, is it kind of the typical thing, piece of innovation where it's expensive at first and then it slowly gets democratized over time? Or is some of this stuff like really just going to be incredibly expensive? I'm sure there's like an insurance component. What's your thoughts on democratizing access to some of this reproductive technology as it rolls out over the coming years?
Yeah, that's always an important question we have when we are entering sort of like new technologies that some of them can be quite pricey at the beginning, quite expensive. Here's sort of like my a cynical view, and also I want to kind of like also talk about why I think that probably won't be the case, but we can't keep our eye off the ball, so to speak. So for better or for worse, without sounding too dismissive here, generally speaking, the entities that are aiming to solve some of these important problems are also for profit. In fact, I think the more accelerating science happens, it's like more for-profit environment. And because of that, it's kind of like in the interest of these entities to, generate more profits when they cater to pretty much everyone rather than just to a select few. So I think from that perspective alone, and given from the historical way in which we've adopted, medicine is there has been, maybe apart from a few elective surgeries early on when they are a lot more expensive, less affordable, where you see a certain, more the wealthy individuals perhaps go more for it than your average person who has a certain budget that can deviate on. But for the really important, like life-changing, health-changing medicine, they are, in a subsidized way, they are very, very democratized. I can't really think of any major examples where they are out of reach for the average person. So I would like to say, from the more cynical perspective of for-profits, which drive a lot of some of these decisions where, they are distributed to more to the public at large. But I think a more optimistic view, right, which is as a species, as I've said, we've tended to do the right thing, which is to make this obviously more accessible to everyone because it helps everyone. And I think we've done that, and I want us to continue to keep an eye on it and not deviate from the path. But that's actually where I see this going as well, which is, really people will think about the benefit for everyone, not just for the select few. I can't think of any major examples that are not sort of like more elective, sort of like aesthetic surgeries that people do. For more fundamental health related stuff, it's a lot more accessible to the average person as well.
Right. Okay, great. So we have a good number of questions coming in here from folks who are tuning in. So I want to transition to those. I have plenty more questions I would love to ask you myself, but let's get into some of these audience questions. I'll go through them in the order that we've been receiving them. The first one here is, are you familiar with Dr. Shauna Swan's work regarding male fertility and sperm count decline over the last number of years? If so, can you comment on that?
I'm not particularly familiar with that person themselves, himself or herself. I'm not exactly sure here. But of course, the concept of low male fertility and low sperm count has been around for a bit. It's also part of sort of like some of these more fundamental environmental changes that are happening in reproductive and fertility rates across the globe. So there's actually a company that's been funded. I mentioned in vitro gametogenesis, and I mentioned making these germ cells, egg and sperm, in a dish. Most of these companies are more catered to female reproductive because usually eggs are in shorter supply. But there are companies, and there is this particular company, I think the name is Paterna, that's recently been funded to solve in vitro gametogenesis for sperm, pretty much, and so to make more sperm in vitro. So it's been, at least from the IVG point of view, it's being tackled. But so like a lot of these like developments in a lot of these technologies that we have discussed will help there quite a bit as well.
When using embryonic stem cells, do you have to design or code the cells to what you want, like i.e. gender, or do they do that automatically?
So there's one thing that, I guess I can explain a little bit about the biology and some of the implications for using embryonic stem cells, which come from potentially the somatic cell you're deriving them from, and then they'll be used for deriving these gametes or these the sperm and egg. I think one of the questions I actually get is, well, wouldn't it be kind of like a clone because you only have like one set of like chromosomes pretty much to work with? And there's this cool thing that happens during the process of oogenesis or spermatogenesis, where all our bodies have two copies of chromosomes in each cell, but your germ cells have to have only one copy because it will allow for one set coming from a mother and one set coming from the father. But during the process of splitting to make these what are called haploid or one set chromosome cells, there are these events called crossing over, where a lot of genetic information is exchanged between these sister chromatids, the chromosomes, when they are condensed. there's also room there to make more changes by editing. That's also a topic that we're not necessarily exploring, but I think, going back to your question about what you learned at Colossal, I learned that you can do editing a lot easier in embryos. Like, are we ready to have that conversation, pretty much? And so you can engineer diversity, genomic diversity as well when you're in the process of generating embryos. And so, yeah, with human ESLs, you're limited. Mostly you can just make pretty much X germ cells. So that means, oocytes, ova. But with male embryonic stem cells, because it contains an X and a Y, you can make, you can make both kind of germ cells, egg and sperm, and therefore why you would have things like bipaternal embryos and so on. So I don't know whether I covered a little bit. I don't really know where the question was necessarily fundamentally heading, but pretty much you can engineer. There is already an innate way or developmental way to engineer genetic diversity, even though the in vitro gametogenesis starts from just a set of or just a genomic set. So you want to diversify the genome so that you can make progeny that look different, have strike a variety of genetic outcomes. You can also engineer it. And also these embryonic stem cells will inherently contain like the power, the potential to go both ways, to have both, to develop both sperm and egg.
And our next question is very related to that. Does that mean that you can engineer or edit to remove, genetics that you don't want, whether that's hereditary diseases or whatever it may be, for children in the future?
Yeah, absolutely. Again, these are moratoria that's been called even more recently in terms of germline editing, but
Mm-hmm.
I think I'm ready to have that conversation as well, because a lot of the monoallelic or Mendelian inherited diseases, and even the polygenic, more complex diseases like diabetes, autism, and so on in the future, can be corrected by doing germline editing. Of course, in an open, transparent way. We obviously have to have those conversations and see whether it's really what we want to do. But in addition to advancing the technologies of better screening, right? If you have more embryos to screen, potentially you don't even have to edit. So better screening would
Mm-hmm.
obviously help quite a bit, because you have a limited embryos, but still there's more than one, and you can actually select for the better embryo by having like the best genetic test out there. But in addition to that, if you really want to increase the chance of eliminating some of these diseases, you don't have to treat later on, it's by doing like germline editing. So and as I mentioned, I think given that a lot of these diseases are just single point mutations, so just a change in one nucleotide or one base in DNA, they can be corrected with a lot of these very cool new genome engineering tools that are mostly CRISPR based. Of course, you probably have all heard of CRISPR. And you can be a lot more straightforward to do in an embryo than later on when the body has trillions and trillions of cells.
So this next question is about embryos and their ability to rejuvenate and heal. Is there any research or work going into trying to figure out how to replicate that in aging humans?
Yeah, so now, of course, I'm not like necessarily a developmental biologist, but there's a lot of rejuvenation and a lot of also trimming that's happening as you develop fully into a more mature human. And I mean, not even like humans and babies. We're talking about other species that have the potential to rejuvenate. And of course, it's being explored. You can always think about like the biological work or the cool biology work you did as a high school student, right? Trying to rejuvenate the limb of a salamander and so on. So how do you port those systems into like an adult human? That would be interesting. But yeah, I think this process of induced pluripotency, so going back in time, it's already a very, very cool platform to build on. Hence why we built that, we built the platform to rejuvenate bodies by expressing this set of transcription factors, doing in vivo reprogramming. So resetting the epigenetics a little bit further back in time. So yeah, just by the process of induced pluripotency, we found quite a bit of that pathway that can be used for rejuvenation. There's, of course, more more to discover there, and either through individually studying that organ or just the you know, the organism as a whole. But yeah, we have a lot to learn. But I think the in vivo reprogramming approaches using you know, these transcription factors to derive embryonic stem cells that can be used for rejuvenation in an adult, at least in an adult mouse, right? It hasn't been fully tried in a human body yet.
Mm-hmm. Uh, on the same in the same vein of the rejuvenation topic, there are some questions on alternative medicine, acupressure, and its ability to rejuvenate cells. Are you aware of that, or is that kind of within your wheelhouse at all to comment on?
Probably not my wheelhouse. I mean, I have
Okay.
thoughts on some of them. I don't think I necessarily am the subject matter expert there, so I'll probably leave it to people who have explored those more. So like my expertise is mostly sort of like stem cell technologies and what we can do using those in the future. The rest is more like personal stuff, like
Right.
personal, yeah.
Right.
But as I said, like I mentioned a couple of things, like at least platelet-rich plasma, which, you know, you probably are aware a lot of people use for sort of like skin rejuvenation in the face.
Mm-hmm.
That similar concept has been used to, you know, to just rejuvenate the ovaries in vivo. There are like a couple of clinics that can do that. You can try. They purport to have success with that, to extend reproductive age using those techniques. And as I said, I think there are a few companies also in the rejuvenation space trying to explore mitochondrial transfusion. I mean, aging is also like an energy, fundamentally an energy problem. And, you know, mitochondria over time, you lose the number, you lose, you know, the genetic, a lot of the mitochondrial DNA will accumulate mutations over time. So can we reverse that as well? And so some of these like mitochondrial based rejuvenation companies are working towards that. Can you, you know, can you pretty much transfuse mitochondria to reverse aging? So that can be used obviously for reproductive work and increasing fertility, you might imagine. And
Mm-hmm.
I'm personally also interested in that space as well. Yeah, so those are some of the things that come to mind. In terms of outside of that, I'll probably leave. That
Yeah.
would be a cool new topic for your next
Yeah, there we go.
discussion. Yeah.
Um, you mentioned the impact of AI on this space. Is quantum computing and those advancements in your thoughts or the scientific community of biotech at the moment, and the impact there?
I mean, computing for sure, and I'll leave like whether it's quantum or of any kind to the experts in the field. But I think what I'm really interested in as well fundamentally is sort of like these different kind of reproductive paths that evolution has taken us through, depending on sort of like the species in question. And it would be very, very cool fundamentally to understand comparatively. You know, we look at comparative genomics to understand what species. What makes a species different or similar to another species? Can we input a lot of data to also understand how the different reproductive systems are like diverge even within mammals. You know, some you know, in addition to just different placental types that some of these mammals have, you know, they can carry different number of progeny. Can some of those pathways be ported in one way or another? And we really don't know because the network is a little bit more complex. So I think AI can really help us there, where we can input a lot of the data, genomic data, hopefully epigenetic data as well from these organs in question, such as like the gonads, and understand, can we tweak it? Can we developmental tweak it? In fact, there is one thing that actually I would like to say here that, again, it's a little bit tricky to navigate as a topic, but I'm gonna still, you know, just put that thought in your
Let's
mind.
go.
We can really think about in the future whether we want to pursue or not. But we really want to understand fundamentally the process of development and try to understand sort of like more developmental or universal developmental features across species. And of course, again, AI can help there because there's so much data to analyze. There's so much omics data, proteomics, you know, metabolomics, genomics, and so on to input so that we can get like a fuller picture of how different species, even within species, but also across interspecies, how things are similar and different. And if you understand that process well enough, you might be able to also tweak it. And of course, you can tweak it in terms of some of the, you know, traits you can engineer, right? So engineering outwardly traits like hair and so on. Of course, you can see different animal models out there with like different, you know, hair traits, morphological traits and so on. It's still somewhat still superficial, and we can do so much more work there. But developmentally speaking, I'm interested in actually one thing that I would like to see maybe being explored in the near future or later future is, can you make a biological incubator? I mean, ultimately, nature does it best, right? So can you tweak it well enough such that you don't necessarily, so in addition to making a completely mechanical device for gestation, if we're going the mechanical way, can you make a biological incubator where you don't develop the parts of the body you don't need and only develop the parts of the body you do need? So in this case, you'd recreate the full reproductive tract to gestate from that same individual using their stem cells to gestate their progeny without having to necessarily try to recreate,
Right.
Electromechanics and so on, or fluidics. So that's kind of interesting. So can you remove sentience from a species that it becomes sort of like also an incubator potentially for your own embryo in the future without the parts that kind of give us consciousness as sentience and pain sensation and so on. So that's an interesting concept to me, but you do have to understand the biological process right away. But I would give you an example. There's mice out there that you knock out this particular gene called lim1 and they become headless. And so, fundamentally, our genetic blueprint does have the program where you can tweak. We just
They
have
become
to.
headless as like their whole body doesn't have a head, or they don't have like a
There's a
working
little bit
brain.
of a remnant of a head, but pretty much, yeah, they are.
Wow.
Yeah. So, you can consider it weird. You can consider it like
Right.
whatever that may be, but that actually gets to the point of what are the processes that get us to the body morphology we have, and can we tweak it enough such that it serves a particular function without and bypassing the ethical implication of using, because I think using a human surrogate actually is a little bit more ethically challenging than potentially using a biological incubator that is not sentient, right? We are
Right. Is a
actually relying
recreation.
on a human, and human surrogacy is just has really, really taken off. People pay for that process. And in my opinion, I think we shouldn't do that. We probably should. I mean, obviously people are open and they should make the decisions they want. But this I find would be better alternatives ultimately.
Yeah, very interesting. I'm sure an unsettling concept to some, and maybe an exciting concept to others, and but a
Oh, that was.
very interesting thing that is becoming within the realm of possible scientifically, and so something that's worth discussing at the very least, because yeah, things are getting wild, and of course I think it's worth noting, like, it's wild to us, to our kids, maybe things like this will be normal, and that's just how the generations adapt as you go on.
Have those conversations. As I said, so society, we've always moved towards one direction or another, and
Mhm.
generally in the right direction. I want to be mindful, however, that it's good. It's also the onus should also be on the naysayers to say why we shouldn't make the progress we could be making. It's not only on the people who are making the progress to show the benefits. We also have to put the conversation and the pressure a little bit on the people who really want to put the stop on these, because they really have to, given that a lot of the things that we want to do have major benefits, right? So
Mhm.
they also have to explain why they're against these major benefits and this major progress. So the conversation has to be like the pressure has to be on both sides, where people are more skeptical and conservative, as well as the people who are a bit more progressive with some of and forward-looking with some of these thoughts. And as long as we have those open conversations and transparent conversation, then let society make the decisions they need to make.
It's an excellent point. Keeping that tension across all exponential technologies is very important to try to achieve the best possible outcome, uh, because they are all very powerful. Another question here is for anyone who's more senior and never had the chance to save embryonic materials. What options might exist to save DNA for future health needs?
Yeah, so I mean, there are some of the companies out there, for example, that like save the umbilical cord or freezing material. you definitely can, you can definitely freeze your, whether functional or not, your sperm and egg, and they do contain genetic material. I probably have to look into the space of just saving any material, just saving blood, you know, outside of just umbilical cord blood, which used to be sort of like a major thing back in the days, you know, to create enough stem cell for body rejuvenation later on. But with the concept of in vitro gametogenesis, for example, I think that can be quite powerful, because essentially you are rejuvenating. First of all, like any cell type can be rejuvenated through this process of induced pluripotency, right? So you express those core factors, bathe them in particular a cocktail of media to bring them back in time. And that can be done with an 80-year-old and 90-year-old as well, not just with young individuals. I think that's the beauty of the technology. And with the power of genome editing, you can correct those the mutations that accumulate with age over time, because you know, our cells continuously divide and continuously propagate, and the process is not always perfect, and so they're accumulating mutations. So just rejuvenating a cell with an incorrect or a barren genome might not be in your best interest. So, but you do have the tools to correct them. So, provided you're alive in some way, you can always donate the material to, so like, you know, or you can use that material for these technologies at any point in time. But in terms of so like for potential posterity, yeah, probably, you know, either through some DIY methods, or you know, finding the next company out there that can store your genetic material, I think would actually be pretty valuable, and actually should be more widespread. That can be definitely used. Obviously, if you have the choice, you know, save your young cells over the older cells, but with the technologies we're building in the future, technically it won't matter. You can correct the aberrant genome and you can rejuvenate the cell. So as these technologies get better and better, that wouldn't be a concern.
Right. I think we have time for one or two more questions here. This one is a longer
By the
one.
way, I do have to say, but I'm sorry, I
Go
actually do
ahead.
have to say one thing, which is also like pushing a little bit of the limits, but provided you have a digital copy of your DNA, you can use another cell type, another donor cell, to engineer your unique individuality, even, you know, in the future, and recreate your genome. Yeah, epigenetics is a little bit more complex, but at least your genome completely, even using like a different, like any other, you know, human cell, or, you know, if we're gonna be like crazy, any other like animal cell,
Right.
provided you have a copy.
And you know, theoretically, life becomes fully programmable at that point.
Correct.
Which is
Save your
wild.
genetic save your genetic code, and I think that's actually like the best start.
The next question is actually, you touched on it briefly, about understanding epigenetic regulation. So not just mapping methylation or histone markers, but actively mastering how environment and cellular memory shape gene expression over time. Could this be the real key to success in reawakening extinct species, or just stating viable life outside the womb?
Yeah, so different ways to tackle that question. So on one hand, of course, as you know, epigenetics plays a massive role. A lot of the environmental effects we feel during development, during life, is imprinted in us through these epigenetic changes. And when we study in biology, when we study a cell and its transitions, even from like one cell state to another, we don't usually we don't just keep note of what's happening, because generally the genetic component doesn't change too much, other than potentially accumulating some of these you know, random genetic mutations. But the epigenetics is massively important, because they are truly what makes a cell different from another cell. Like what genes do you have on versus off? And there's a lot of omics data of different organs, different cell types out there, and we can obviously collect more. So that can always be used for, you know, AI-driven knowledge build, bioinformatics analysis, comparative analysis, and so on. For extinct species, it's a little bit interesting. I think for some of the more preserved species, I think you can understand a little bit more. Some of that is still preserved, like DNA methylation. You can also find some DNA methylation changes that are different between, let's say, Neanderthals or ancient humans and modern humans, because you actually have that DNA methylation knowledge, or DNA methylation information that can be accessible. For more histone, you know, these like tiny protein cores which are used for wrapping DNA around it, and they get to be modified, and they have sort of like different kind of modifications that result in some genes on, some genes off. It's a little bit worse to find out the information there because, as you know, DNA is a little bit more long lasting than protein is. So it's a little bit more challenging. I think ultimately, I think a lot of in my opinion, the majority of what makes a species similar or different is fundamentally through DNA. And so if you recreate the genomic component, I think the environmental epigenetic stuff gets like layered on over time, depending on where they are gestated, where they are raised, and so on. So yeah, fundamentally, I believe more on sort of like the genetic component is still a bit more dominant. But
Right.
the behavioral component coming from some of this epigenetic layering, that's a very, very interesting question to tackle, and you wouldn't know until you actually have the species to study.
Right. All right. We're at the top of the hour here. I know we're out of time. I want to give you one more kind of wrap up question here that's very much out there, but if your field achieves, your most ambitious goals, what does human reproduction look like in 2050? So in 25 years from now.
It's hard to make these kind of predictions. The technology is there, and I think ultimately we've always pursued these cutting edge technologies for our benefit, and they are obviously very beneficial to us. I think we'll probably move a little bit away from, one of the things that are always going to is always going to be a question, at least with emerging technologies, what is natural versus artificial? Like you put these things in two buckets. And
Mm-hmm.
especially with these trends come and go, but it's trendy right now to think about more of a sort of like the natural way. That used to be the case, of course, when the technology of IVF was developed. It was very stigmatized. It wasn't the natural way, right?
Mm-hmm.
But very soon it was so beneficial. So it changed so many lives that people don't think twice about, right? That it is an artificial way to make an embryo early on. But we don't really put too much thought into that. So I think the divide between what's natural versus the artificial and sort of like potential stigma that goes with it will probably dissipate over time. That's like at least one thing that I feel like we can predict, which is maybe we can feel a certain way now, but over time, as you see the benefits of some of these technologies, that line that it has to be, it has to be all natural versus all artificial or like a mix. I think it blurs and we get less reliant on sort of like that. Another thing is
Right.
these technologies can also can give you more freedom because it removes so many restrictions. At the end of the day, we can still continue as a species because you can generate so many more humans potentially from humans who can, because maybe you don't have to have just one child, you can have 20, and it's possible. It's more, you're going to have only an economic constraint because biologically you probably will not be constrained with things like ex utero technology. And so you can sort of like give more of a breathing room to people who actually absolutely do not want to have that choice for themselves, but will still propagate as a species. So we'll see hopefully the propagation of our species, but without necessarily like these constraints we have, and potentially even making sort of like life interplanetary because we will able to sort of like genetically engineer the future humans. Those are some of like thoughts that come into my mind. Just like some of these more isolated communities like the Amish or certain indigenous tribes, there will always be that some of the in some way groups of society that still want to keep it more sort of like a natural progression of biology and
Mm-hmm.
never be sort of like scorned or discriminated. It's a way
Right.
it can coexist. So I would like to see more of that coexisting happening in the future. So whether it's like completely using more of these artificial ex utero technologies in vitro gametogenesis versus like more sort of like natural approach to reproductive fitness.
Right.
There is no just one path, in my opinion. All paths should be explored.
Exactly. An incredible amount of diversity in the human experience and room for all of it.
But I do have to say, like bringing it back, from some of the work that I did, or at least I was familiar with in conservation biology, I think the most important thing is let's not wait until the last minute to enact change. We have to be proactive, right? You have to like you have to be the change now, pretty much. Yeah.
Yes. Love it. That's a perfect, wonderful point to end on. I want to thank you Eriona, for being here. And thanks everyone for tuning in, for all your questions. Really appreciate it. We will follow up within a day or so with the recording, as well as a recap and some additional resources. And Dr. Hysolli, where can folks find you if they want to follow you, or are you on social media or anything?
Interestingly, I try to stay away from social media, so I'm not really
Nice,
aware, but I'm on LinkedIn.
nice.
Feel free to
All
connect.
right. All right. Sounds good. All right. Thanks, everyone. Have a wonderful day. Thanks all for being here, and we'll see you next time. Appreciate
Thank you,
it.
everyone. Bye.